also, I want to let you know I wasn't at office hours yesterday, obviously, but I want to start coming next Thursday. I had some different other plans. So, yeah, just want to let you know. I think my friend Shelly might have went though. So.
Okay. Yeah, there was a decision.
Awesome.
Yeah, no problem. I'm always there.
Awesome.
Usually.
Oh, okay.
Stuff could happen.
Good to know.
It's a little screwed up. But you can also come over. The other thing you can do is come over. I've got to say, Okay, let's see. Microtubules here. Let's see. Okay, so we went through pretty much the basics about dynamic instability and looking how microtubule polymerization, depolymerization is regulated predominantly due to rates of GTP hydrolysis on beta-tubulin. All of the actions pretty much at the plus end. Let's see. Went over a couple proteins that are MAPs, microtubule-associated proteins, that control the spacing of microtubules and axons. So we're going to pick it up and continue discussing some other proteins that can modulate microtubule dynamics. And then we're going to switch over to finish up the unit by taking a look at motor proteins and how they function, using microtubules as an infrastructure to carry vesicles. Okay, so one protein here, it's another microtubule-associated protein that's kind of funky because it also can be used as a carrier protein. And this is called a tip protein, TIP. It's a plus-end tip because you always see it at the tip of a plus end. And it's a little protein called EB1. And EB1 has two important functions. The first is that remember when you've got a microtubule that's polymerizing, keeps going up to here, and then it undergoes a catastrophe, shrinks back down, it gets rescued, grows again, another catastrophe, rescue. One of the questions that was asked was, well, how do you know when to rescue a microtubule that's shrinking? Well, it turns out the guy who plays the largest role in mediating rescue is this little guy here, EB1. And remember that what's happening when a microtubule has undergone catastrophe is that all of those protofilaments are loaded with GDP beta-tubulin. And it changes the structure of the tubulin in the protofilaments so that they have a curvature. And they're actually curving away from each other, okay? So the trick in getting them to be rescued, and this is what EB1 does, is that EB1 binds, it grabs on to the plus end of a shrinking microtubule, and what it literally does is it forces the protofilaments to stay together. It's almost like a molecular glue. And what it does is it forces those GDP-containing tubulins not to continue flying apart from one another. So it holds them together. And what that does is it stops further depolymerization. So it holds that shrinking microtubule in place. And now what it does is it allows it to now start growing again. So it sort of stops the depolymerization, and now new tubulin can bind to it, and now you can start growing it again. So that explains the rescue part. But the other thing that's interesting about EB1 then is that it doesn't dissociate from the now rescued and now regrowing, repolymerizing microtubule. Instead, what it does is that as you start rescuing this microtubule and you start bringing in new tubulin, EB1 moves from the tubulin it was on to the next one. You then add a new one, it binds to the other one. So what it is actually doing is it is constantly translocating and remaining bound to the most recently added tubulin on that microtubule as it's being rescued. So in essence, what it is doing, it's catching a free ride. It's not a motor. It's not walking. It just remains bound to the most terminal plus-end tubulin that gets added. So in doing that, what it does, it's also ensuring the microtubule keeps growing because it's keeping the protofilaments held together. But what it's doing is catching a ride along the plus end of that microtubule to wherever it's going to go. Which, again, since the plus ends radiate out towards the periphery of the cell, it means it's generally headed towards the plasma membrane. Okay? cells take advantage of EB1. And that is, it provides an alternative mechanism to carry stuff to the end of a growing microtubule. You don't always have to use a motor to carry, for example, a vesicle or an organelle to the very plus end of a microtubule. You can hitchhike, and generally these are smaller than vesicles. They're not going to be vesicles, they're going to be other proteins. So the cargoes here are proteins that you might want to carry out to the plasma membrane. And they literally hitch a ride on EB1 because as EB1 gets carried out on this growing microtubule, you carry your hitchhiker along with it. So it's another way of taking advantage of a growing microtubule to deliver a cargo out to the end. And you can actually see this in a video clip. It's really pretty cool, where what you're looking at here are cells that are expressing GFP tag EB1 or other plus-in tip proteins. Okay? And what it allows you to do is actually watch the microtubules grow towards the plasma membrane, because what you're actually seeing here is the fluorescence of the EB1. So for one thing, what it really illustrates is how dynamic the microtubules really are. I mean, you can really get an appreciation because every single one of those little fluorescent traces that you're seeing moving out to the plasma membrane, that's a microtubule that's growing. Okay? And what you're actually seeing there, the fluorescence, is all due to EB1. All that fluorescence is on the very, very plus end of the growing microtubule. Okay? So you can really get an appreciation of how dynamic it is. You can see how rapidly this is taking place, but you can also see how well they're all moving out to the plasma membrane. So again, it's just another mechanism that cells take advantage of to deliver cargoes to a destination, and they can do it in an alternative way without having to use. So you're basically using the growing microtubule itself to carry stuff out to the plus end. Okay? So it's just kind of a little cool sequence there. Okay, a couple of other proteins. This is a motor protein that's very unusual. It's called kinesin 13. So as you're going to see in a minute, kinesins are plus-end motors. They move towards the plus end of a microtubule. Most of them, the prototypical kinesin that we're going to talk about, kinesin 1, just carries stuff. Kinesin 13 is unusual because it doesn't necessarily carry stuff, but what it does is it actually promotes, as it's walking towards the plus end of a microtubule, it's pulling up the protofilament that it's walking on. So every time this kinesin takes a step, it's pulling the protofilament back. Now, as you're going to see, motor proteins have to hydrolyze ATP with every step that they take. All right? It's very energetically demanding. So as kinesin 13 walks towards the plus end of a microtubule, it's pulling back the protofilament that it's moving on, and it's actually promoting depolymerization. Because what it's doing is it's peeling back the protofilaments so they look like they're all going to be loaded with GDP. So it's exacerbating that curvature, and it promotes microtubule depolymerization. And where you're going to see kinesin 13, this is just kind of a preview. Kinesin 13 is going to play a role during mitosis. And it's going to cause microtubules to become extremely unstable while they're in the process of shooting out from the centrosome and trying to capture chromosomes. So it basically has a function during the formation of the mitotic spindle. So microtubules are spewing out from the intoc, trying to lock on to its binding site, which is going to be called the kinetochore, on the chromosome. It's a little tiny target. So most microtubules that are coming out, they're trying to hit the bullseye. And it's a really, really tiny target, so most of them miss. What kinesin 13 does is to make sure the microtubules that come out and miss depolymerize very rapidly, go back to the beginning, so you can spew out more of them again. So it plays a really crucial role in causing microtubule dynamics to become even more unstable when you go into mitosis to ensure all your chromosomes get captured. Okay? So just a heads up that you'll see this guy later. The other one is a protein called staphmin. And staphmin is sort of like thymosin beta-4 was for actin. What staphmin does is it binds to tubulin, and it's a gap. And so what staphmin can do is it binds to the plus ends of microtubules. It acts as a gap that greatly accelerates GTP hydrolysis on beta-tubulin. So it's going to accelerate depolymerization. So again, it carries out a role in promoting instability, causing microtubule depolymerization. But here's where it has another function. And that is it remains bound to a pair of adjacent tubulins. And what it does is it keeps these from being reincorporated into another microtubule. So it not only causes depolymerization to take place, but it's actually grabbing the tubulin and says, you can't be recycled, you cannot be reused. So again, staphmin plays an important role during mitosis where microtubule instability has a very, very critical role. And again, as you're going to see, that's why drugs like Taxol that stabilize microtubules are antimitotics, because they prevent this dynamic instability from happening when cells are going into mitosis, and also, most importantly, to move your chromosomes during anaphase. Okay? All right, enough of the modifiers. Now we're going to get to the motors. Okay? So we have to take a look at the problems that you find if you're trying to use microtubules to transport vesicles or organelles in neurons versus non-neuronal cells. Okay? If you take a look at a non-neuronal cell, all the minus ends are embedded in the centrosome, the end top. They all radiate out with their plus ends towards the plasma membrane. That means you've got a polarity set up on the inside of the cell. So what if you take this and now you superimpose it over here? Here's the secretory pathway now. So now you're looking at the relative locations compared to the centrosome of all the compartments in the cell, for example, that you're generating vesicles and have to move them. Okay? So for example, here, if you've got the centrosome, we've got the endoplasmic reticulum, here are the Golgi, and now we've got secretory vesicles that are coming off of the Golgi and having to move towards the plasma membrane. The only way you can transport those vesicles towards the plasma membrane is you have to use a plus-in motor, because the plus-in is all the way out here. You can't use a minus-in motor. So the plus-in motors that are going to be directing vesicle traffic towards the plus end or the plasma membrane, it's going to be referred to as anterograde transport, forward transport, and it has to use a plus-in motor, which will be a kinesin. Now, what about the opposite? What about, for example, endocytosis, okay? So that now, instead of having a vesicle move towards the plasma membrane, let's say that you've endocytosed the LDL particle and the LDL receptor. You're bringing that vesicle towards the endosome, and now you want to take it to the lysosome. Well, now you're moving towards the centrosome. So now what you're having to do is basically move it in a minus direction, towards the minus end. That's referred to as retrograde transport, and that's going to require a dynein. So kinesins take you out, dyneins bring you back in. Okay? So that's in a non-neuronal cell. Now if you take a look at a neuron, it gets a little bit more complicated. And the first thing that you notice is that in a neuron, microtubules get sheared off. They don't all remain connected to the centrosome. So neurons have fragments of microtubules. Now they can still be very, very long, very large, but you can't trace all their minus ends down to the centrosome. Now, one distinction, though, with neurons, relative to a non-neuronal cell, these microtubules are very dynamic, constantly growing and shrinking. You look at a neuron, microtubules are essentially very stable. The half-life of a microtubule in a neuron is the same as the half-life for the cell. They're basically permanently there. But what you notice is the following. We've got a bit of an unusual situation. If we look up here at the dendrite, okay, so that's the part of a sensory neuron that's going to perceive the stimulus, temperature, light. What you notice is in the dendrite, the microtubules are arranged randomly. Some of them have their minus ends out towards the dendritic terminus. Some of them have their plus ends. So they're facing both directions. So what that means is out in the dendrite, you could use either a kinesin or a dynein to carry your traffic in either direction out there because you got microtubules that face both ways. Kind of simplifies the problem there. When you look at an axon, though, and here's where the problem becomes more acute, because axonal transport, axons can be very long. Again, the length of an axon in your sciatic nerve that would run from your spinal column down to your foot, a single axon can basically be 1 meter in length. That's pretty long. That's a very, very long distance that you have to transport neurosecretory vesicles down the length of that very long axon. The transport going towards the axon terminus, all the microtubules, even though they're in fragments, they're all oriented with their plus ends down at the axon terminus. The minus ends are all still pointing back at the M-top. What it means is this. A kinesin that's going to be carrying a neurosecretory vesicle towards the axon terminus, combined to this microtubule, walk down here, it's going to fall off, and it jumps on another one, walks to its plus end, jumps off, walks down. And in essence, what the kinesin is doing is it's having to translocate from one microtubule to another to ultimately make its way down to the very end of the axon terminus and get there. But they're all moving the same direction, okay? So axonal transport involves a longer distance than you're ever going to find in a non-neuronal cell or what you're going to find in transporting vesicles to and from the dendrites. So axonal transport was really the system that was first exploited to discover motor proteins. All right? To give you an idea, though, of this type of transport, okay, so here's GFP. These are GFP-tagged secretory proteins that are moving in a non-neuronal cell. Okay? So this is just GFP with a signal sequence, and you can basically follow. You can see a little bit of retrograde transport, but you notice most of the vesicles are all moving down the plus end towards the plasma membrane where they're going to be secreted. And you notice it's kind of a herky-jerky movement. Well, there's a reason for that. Those microtubules are very unstable. So you've got a kinesin that's taking a vesicle down the length of a microtubule, and the microtubule can be depolymerizing in front of it. Literally, the road is being cut off from underneath it. So what does it do? It just has to hop onto another microtubule that's growing and switch off and continue walking down that one. So the difference here is that unlike a neuron, what vesicle trafficking in these non-neuronal cells are having to deal with is that the microtubules that they're working with are shrinking and growing at the same time that they're moving down towards the plasma membrane. Okay? So it's dynamic movement occurring on a microtubule that itself is also very dynamic. Okay? All right, I want to show you this one. The clip doesn't run embedded in PowerPoint. So what I want to show you is what these look like. These are melanophores. Melanophores are pigment-containing cells in a number of organisms, particularly amphibians or fish. And what they can do is that they change the concentration of melanin pigment granules to either aggregate them in the center of the cell or to send them out to the periphery of the cell. And it's a way of camouflage. It's a way for these organisms to change the density of their pigmentation so that the cells can adopt and become lighter or darker in response to whatever background they're found in. It illustrates anterograde and retrograde transport beautifully. Okay? So you're going to see an example of each of them here. These are in cultured fish melanophores. But I just have to pop it out of PowerPoint here to show you guys. Okay, here's one. And there's the other. Yeah, so the one thing that you can see is that if any of you guys have observed amphibians or fish, they can do this very, very rapidly. They can change their pigment distribution and their coloration very, very rapidly, depending on if you put them in a dark background or a light background. And the reason they can do it that quickly is that they're moving the pigment granules, like in here, this is retrograde transport, pulling them all back in from the periphery of the cell towards the intox. That's all being driven by a dyning. So these are dyning motors that are pulling granules and concentrating them in the middle of the cell. That cell's going to look very dark and pigmented because they're all concentrated. Here's a cell that what you're seeing here, you're dispersing all of the pigment granules out to the plasma membrane. So that's using a kinesin motor, okay? But the one thing that you can appreciate in both of these cases, how quickly this is taking place. And it's all the more remarkable when you realize these motor proteins are carrying these vesicles one step at a time. They're taking sequential steps down the length of a microtubule. But when you look at it, it's seamless. It's basically just as if the vesicle's being transported along the length of a microtubule, but that really shows you how quickly that's taking place. Okay? Okay, and here you can actually look at it. You can actually see these vesicles moving now. And this is the experimental system that was used. These experiments were initiated back in the late 1980s into the 1990s at the Marine Biology Lab at Woods Hole, Massachusetts. It's off Cape Cod. And one of the organisms that's very well suited to studying this, and you can also eat them if you're into calamari, are squid. And so it turns out that within these small squid, what you have is that each one of these guys has a single large axon. It's called the squid giant axon. It's a single cell that's an inch or two in length. And that's the experimental system that was first used to discover motor proteins. Okay? And the starting point that you could do using this beautiful system is that the axons itself are transparent. So here you can actually look, using light microscopy, you can actually look inside the axon, and what you're following there are vesicles moving on microtubules. So you can actually see and visualize the movement. Now you see these large things, they look like little worms. Those are mitochondria. And what is happening in an axon is that the distance that the motor proteins have to move is so great, and they're expending so much ATP. Every step requires ATP hydrolysis. So to move a vesicle down the length of an axon, a single motor protein is going to consume a massive amount of ATP. You need to bring the gas station along with you to provide a very high concentration of ATP to keep the process going. So among the other things that these motor proteins are carrying along the length of these microtubules in an axon, they're not just transporting vesicles, they're bringing mitochondria. So in essence, you've got ATP being produced in the mitochondria and pumped out of the mitochondria along the length of the axon so the concentration is high enough to keep powering these motor proteins. Okay? So here's the thing to imagine. You can actually sit here and you could dissect out, in the course of an afternoon, a very, very large number of those giant axons. Think of it as like a tube. It's very, very, very small. But if you wanted to reconstitute vesicle trafficking, you need to set up a biochemical assay. So your biochemical assay is going to be the following. First, you purify microtubules. You can do that from any tissue, any source. You purify tubulin, you can polymerize it, you treat it with Taxol. So now what you have are Taxol stabilized microtubules. Okay? What temperature do you want to keep them at? 37. So you don't want an ice bucket. You don't want to put your microtubules in ice because they'll depolymerize. So you've got a tube of purified microtubules. You purify vesicles from another cell. So you can do that by density gradient centrifugation. You can purify a bunch of vesicles. You take vesicles, add them to purified microtubules, what happens? Nothing. The vesicles don't bind, they don't move. That's your starting point. You realize you need something else. Well, the first thing you might think is, well, I need ATP, I need some energy. Okay? So you throw in ATP. So now you got ATP, purified vesicles, purified microtubules, and nothing still happens. That means there's something else there that you have to purify, you have to isolate. And it means that whatever that is, it's not stably associated with the vesicles. The vesicles don't have it. That was the starting point to purify and identify the motor proteins. And the way that they did this was really elegant. You take that giant axon, about an inch long, and it looks like a tube. It's much smaller. But what you can do is you can actually take a glass rod. So imagine here's the axon. And what you can do is you can take a glass rod and you can squash this cell. You can squash and push the axon and extrude a couple of microliters out of the end of it, this goo that was inside the axon, and you call it axoplasm. It's basically everything that's inside the axon. That was the starting point to purify motor proteins. So now what happens is you take this crude axoplasm, ATP, Taxol stabilized microtubules, and vesicles, and boom, now they move. So what it meant is that whatever you needed to transport these vesicles on microtubules was contained within the axoplasm. So now what you could do is that then you could discover these motor proteins were also in non-neuronal cells. So you didn't have to do your biochemical purification from just axons. You could pick any cell type you want. And that was the basis for the purification and the identification of all the motor proteins that we now know. Okay? And it turned out there's two distinct classes of motor proteins that are there. The kinesins and the dyneins. Okay? The prototype that they first discovered was this guy here called kinesin 1. It was the prototype. And kinesin 1 is a prototypical plus-in motor. It's got a head that basically binds to the microtubule, and it's got a tail that binds to the organelle or the vesicle that it's transporting. It only moves to the plus end. But they also discovered that there was a different class of kinesins that only function during mitosis. And these are bipolar kinesins. They don't have personality disorders, but they're bipolar in that they've got two heads. And what they found with these kinesins, and this guy here, kinesin 5, is that they don't carry vesicles. They carry another microtubule as its cargo. And where these guys are going to come into play in mitosis is that these bipolar kinesins are going to be responsible for taking microtubules from the mitotic spindle and moving them this way. It's moving them apart. So the key thing here is the vesicle trafficking that we're talking about right now involves stationary microtubules. When we get to mitosis, though, you're going to see that the cargo is going to be another microtubule. The microtubules are actually going to be carrying each other as cargoes by using these bipolar kinesins. Okay. So here's the structures of kinesins and dyneins. Okay? So if you look at them under the electron microscope, they're very, very similar. They're kind of like mutt and jet. Okay? And what really distinguishes them is that kinesins are very long and lanky. And dyneins are short and squatty. But you notice that what they both have is that they have a common head here at the base, and they're separated by a long, flexible arm or chain. And so what it means is one end of the motor proteins is an ATP binding and ATPase activity head that is actually interacting with the microtubule. The tail interacts with the vesicle. So they're bimodular, they're bifunctional proteins. One end basically binds to the microtubule, the other end binds to the vesicle cargo. Dyneins and kinesins are relatively similar in their mechanism. So we're only going to talk about kinesins. It's a little more straightforward. But understand that the basic mechanism that a kinesin uses is very similar to how a dynein is also going to work. It's just that the dyneins move in the opposite direction. Okay? So, remember back the video that we showed the very, very first class. This is where we're going through now and actually looking at the process of how these guys actually move step by step. Okay? So the first thing to understand, each of these heads can only move if it's binding and hydrolyzing ATP. So there's an ATP-dependent hydrolysis and release in order for the kinesin to move. That's the first part. The second part is kinesins only step on beta. Okay? The distance of the step is from beta to beta to beta. Okay? They don't step on alpha. Okay? It's a series of steps, so you can start anywhere. So you kind of have to look at the progression of if you were going to freeze frame a kinesin sort of in the middle of walking, you can either start like this, you could start like this, you could start like that. So the point is, it doesn't matter where you start. It's a continuous series of sequential steps. Okay? So if we go through the process, it's actually starting up here. Okay? So what we have, and what you have to imagine is at the top of this neck, way out here, that's going to be the vesicle that's being carried towards the plus end on this microtubule. Okay? All right, so here's where we start. We've got the two heads. Here's one on a beta. There's the next beta. It's frozen in place. Okay? This guy has nothing bind to it yet. This guy has already hydrolyzed ATP to ADP. So the front head is empty. This guy has ADP because it already hydrolyzed it. Cool with that. To get the thing walking, to kickstart this guy into moving, what we have to do is that the leading head now binds ATP. That's going to cause a conformational change. And what it's going to do is it causes the back guy now to pivot and swing around. So it was frozen back here on this beta. Here's the beta that the first guy's bound to. And what this guy is going to do is the trailing head's going to take a step to the next beta after that. So it's basically going to do a pivot. And it goes like that. Okay? Now you notice the vesicle hasn't moved yet. So if you take a look at it, it's still right here. So now what happens is this guy swings around and it's going to plant itself one beta tubulin ahead of where this guy is. So this guy moves 16 nanometers from that beta tubulin. Remember the betas are 8 nanometers apart. So this guy goes from here up to there, and now what it's going to do is plant itself. Okay? Now, what this guy is going to do is that the back guy here, this one basically now is going to release ADP. It lets it go. This guy up here is now going to release it, and it's going to, this guy hydrolyzes, sorry. This guy here is going to hydrolyze ATP to ADP, release the phosphate. This guy up here now is going to let go of the ATP that it had. So in essence, what it's constantly doing, every pivot, is that it's hydrolyzing ATP, swinging it around, releasing the ADP, and now you have to bind a new one. So it's constantly alternating between hydrolyzing ATP, releasing ADP, you have to bind a new one. And it literally has to do this every single time that it's making this step. Okay? So here's the thing to bear in mind. Okay? So here's where you have to think about how energetically demanding this is. Each step is 16 nanometers. You notice it also stays on the same protofilament. So these guys actually aren't rotating around from protofilament to protofilament on a microtubule. They're staying on the same one the entire way down to the end of that microtubule. You can have multiple motors carrying multiple vesicles in the same direction or opposite direction on the same microtubule. Okay? So it's walking down that same protofilament. Okay? It's moving 3 microns per second. And here's where you get an idea of the speed. Okay, it's moving 3 microns per second. If you've got a 1 meter long axon going down from your spinal column to your big toe, how many steps is it having to take to do that? So that's the first thing to consider when you're doing it, is how long would it take if it goes 3 microns a second and it needs to travel a meter? How long will it take to do that? The answer is it takes a little while. It doesn't happen immediately. But more importantly is think about how many steps is it having to make to do this, and understand that every one of those steps involves an ATP hydrolysis event. So it's an incredibly long journey, but when you look at it in the video clip, you notice it looks completely seamless. Okay? So it's the rate that's involved here that allows your vesicles to move these very, very long distances. Understand that if you had to rely on random diffusion to get a vesicle to go that far, it would never make it. Okay? There's just too much freedom of movement that they could go. So, now here's the rub. It can't make it all the way to the end. Here's the last hurdle you have to overcome. The microtubules don't go all the way to the plasma membrane. They come up short. So here's the problem. A microtubule will only get you so far. Well, you don't want to rely on random diffusion to take a vesicle that you've transported, say, a meter from all the way down here, all the way up to that point, and then loosen it here and expect it to make it to the plasma membrane by a random drift. You kind of defeated the whole purpose there. So we actually have to have a handoff. Okay? And you can actually see the handoff take place in a video clip. What this little clip shows you is notice the size difference between a microtubule here and a microfilament right there. So that really exaggerates and lets you see the difference in size between a microtubule and a microfilament. We're going to hand off the vesicle from a microtubule to a microfilament. But that means it has to switch motors, because kinesins and dyneins only work on microtubules. The only motor protein that we can use on microfilaments is myosin. So there's a handoff that takes place when a vesicle reaches the end, the very plus end of a microtubule, and falls off. It's going to pick up a myosin, and the myosin is going to take it the rest of the way to the plasma membrane. So here's what it actually looks like. And what you have to do is keep an eye on the one vesicle here. It's going to load up. There it goes. You can kind of follow it. You're going to see it come down right there. It's pretty quick. There's not much of a lag here between when the vesicle reaches that plus end of the microtubule, jettisons the kinesin, picks up a myosin, and then it immediately moves onto a microfilament track, and that takes it the rest of the way. Okay? So the point is at the end of this, what you guys can see is it requires a collaboration between the microtubules and the microfilaments to get your vesicles to their final destination. You basically have to use both to get you to the plasma membrane. The opposite is true for endocytosis. So that means when the LDL particle and the LDL vesicle are first coming into the cell at the plasma membrane, they basically have to hop onto a myosin motor where the plus end is facing sideways or into the cell, and that's what brings the vesicle in far enough to where now it can be captured by a dynein, hop on a microtubule, and now it can head towards the lysosome. So you need both of the cytoskeletons and both classes of their motors to basically move your vesicles to and from the plasma membrane. Okay? All right, any questions about motion now? Okay? We've talked about how the vesicles get to where they're going, how they know where they're supposed to go, what they do when they get there. Now you guys see how they get there. Okay, so I want to spend the last couple of minutes here. Here's what's coming next. Now we're not going to move stuff. We're going to continue the theme of movement, only now we're moving information. How do we transmit information in a cell? Now, what you guys are used to thinking of when we deal with information transfer in cells, you're thinking DNA makes RNA, makes protein. No. We're not going to be doing that. It's too slow. It takes too long. We want cells to be able to respond physiologically very quickly in responses to extracellular signals. Now, you already saw how cells change their mobility, their migration in response to extracellular signals. Now we want to change their metabolism. We want to change what they're doing. We need to do that faster than having to sit around and wait for a gene to make a messenger RNA to be translated into a protein. We're going to modify the activities of proteins already in the cell. We also have to amplify the signal while we're doing that. The extracellular signal we're going to be looking at are going to be present sometimes in the nanomolar concentration range. The physiological output is going to be in the millimolar range. That's a huge increase between stimulus and product. The pathways that we're going to be looking at amplify the signal enormously. Okay? So, where we're going to get underway on Monday then is starting this next section that goes by the name of cell signaling, signal transduction. Our focus is going to be looking at what happens inside the cell in response to an extracellular hormone or growth factor that's going to trigger some very quick changes in metabolism. Okay? All right, so have a good weekend. Go Cubs.